Hello guys, welcome to my YouTube channel, Computational Scientist. Today we are going to talk about we are going to do a tutorial on how you can do protein sequence alignment using ClustalW. This protein sequence alignment will be a pairwise. That means it involves alignment that involves two protein sequence or a multi alignment. That's you have numerous more than two sequencing. Protein sequence, you can do the alignment, calculate the percentage identity for each of these, and we are going to use ClustalW. As we know, there are numerous methods, numerous servers, numerous tools and applications that you can use to do this. But in this tutorial, we are going to talk about how we can use ClustalW to achieve this purpose. Let's go to our browser. Just go to the browser and type Clustal Omega, Clustal Omega, and click on it. Once you click on it, you will see Clustal Omega multi-sequence alignment. At the same time, this is just an online server, and at the same time, you can download the Clustal Omega software on your laptop. But in this tutorial, we are going to work on this online server. Click on the Clustal Omega, and this is it. It's a multi-sequence alignment. Clustal Omega is a new multi-sequence alignment program that uses seeded trees and HMM profile technique to generate alignment between three or more sequence, because it is multi. So when it is multi, it takes at least three sequences. But if it is pairwise, it's just two sequences. So for the alignment of two sequence phase, instead use our pairwise. So if you want to do just two sequence, you use the pairwise alignment tools. But for more than three sequences, you use the multi-sequence alignment. So let's do that. So what you can do now is, if you already have your sequence well arranged in a file, you can upload it from wherever you have it on your local and on your laptop. You can upload that file. But if you don't have it, you can just copy and paste. Let me use example. Let's take these sequences that I'm having here. Let's take like one or two of them. So let's put this. Just give space. Let's take one more. Let's take two more. Just copy and paste. Let's copy and paste. Put space. Let's take one more. Let's use minimum of 3. Let's copy lab. So you copy all the sequence. So once you've done that, you already have the sequence of the 3 proteins, right? So the next thing to do is go to step 2. The first step is copying or uploading your sequence file, right? The next thing is to choose your format output. There are different format output, but you can just choose every one based on your knowledge of each of them. But let's use the ClustalW with character count. So I think we are going to use this with character count, and we are going to try the ordinary ClustalW. So let's try. So once you do that, the next thing is to submit. The first step and the final step is to submit your job. And you can decide to input your email. If you want email notification as soon as the alignment is done, just enter your email, the title, and if you do not want to, it's fine. In this study, I'm not going to put any email, and let's submit. Okay, I think we made a mistake. It says 2 sequences cannot share the same identifier. So there's probably, I think maybe it's this one. Yeah. So let's change. I think that sequence we copied the wrong. Let's copy. Okay. So the problem is we copied this particular sequence twice. That's it. So we have to be careful that you're not copying a single sequence twice. So let's do that, and I think we should be fine now. Let's submit. So it says your job is currently running. Please be patient. Yeah. This just since we are only working with three sequences, it takes few minutes. But if you are using too many sequences, you can just input your email address so that as soon as it is done, because that will probably take much time. But as soon as the alignment is done, you get a notification on your email and a link to access your result. As you can see for this now, the result is already out, and you can see the alignment for each of them. and we can see the alignment for it. So let's look at the result sum. It also generates a genetic tree for you if you are interested. If you are interested, let's go back. I think it only picked two. It only picked two of the alignment that we guess. We're supposed to have three. Let me look at it. 1, 2, 3. So is it the same thing? It's not. Okay, so let's change this. Just be careful in your copy. So one of the best way to do is probably have your file already, and you can just upload your files. So let me check another sequence here. Just copy. Let me have it here. Okay. So let's submit. Let's see. So like I said, it could take some few minutes, and you can set up your email notification. Okay, I think we're done now. So this is the result for the 3. So can you see each of them with their respective identifier, and you can see the sequence number. So let's go. So from this, apart from you doing alignment, there are also some other important information you can get. You can probably, if you want, you can get the colored, the colored two colors, so you can see it gives different colors to different colors, right? So another thing, you can look at the result summary. This is the result summary, the input sequence, choose output. Let's click on this and the choose output. So let's look at the percentage identity matrix. Let's look at the identity matrix for this. Can you see this is the sequence 1, 2, and 3? So what this is trying to do now is it's trying to compute what is the percentage similarity identity between the 3 of them. So the first one is comparing this 1 and 2 to this one. So when we are comparing the sequence of the second one, the first one, 19.58% of the sequence here are present in the first sequence. But for the third one, just 16.96% of it are present in the first sequence. So this way, the second one is comparing this one, the sequence 1 and 3, with sequence number 2. And that's why sequence number 2 is 100%. So if you compare sequence 1 to sequence number 2, we are getting 19.58% of this. Only 19.58% of this sequence are present in sequence number 2. For the sequence number 3, only 18.57% of them are present in sequence number 2. And this third one is telling us that we are comparing the first and second sequence to the third sequence. So if you compare the sequence number 2 with sequence number 3, just 18.58% identity occurs between the two of them. And if you compare the first sequence to the third sequence, just 16.96 of this sequence are present in this particular sequence. Those are some of the information you can get. And another information you can get is the phylogenetic tree. If you're interested in looking at phylogenetic tree, I think, okay, let's go check that one under. Let's check it here. Can you see? Well, because there are two different, so if you have different, this is going to draw the phylogenetic tree for you, and you can probably get your data from it. That's just the summary of how. Then other things is just view results. This doesn't really show. So you can just go back, and so you can download. If you are interested in the result, you can download the result. You can view, and you can do as many things as you want on this. So let's just show color. So this is just a simple step to download, to doing sequence alignment. You can get your sequence from either any protein data bank, NCBI, and protein data bank, the RCS protein data bank, PDB. You can also get it from Swiss Prompt or UniPrompt, depending on wherever you have your sequence. And if your sequence is gotten from the lab, maybe after you sequence your sample and you have some polypeptide sequence, just copy that and put it straight into this software, and this software is going to tell you the alignment, the similarity, and the identity, percentage identity of this, and you can also get the phylogenetic tree result for this, for your multi-sequence. And don't forget, like we show, you can do multi-sequence on this, and you also have another, choose on this same software for pairwise sequence. We are doing sequence for just 2 amino acid sequence. Thank you very much. Please kindly watch, like, and subscribe to this channel so that you can get more updates on different techniques and tools for bioinformatics and computational studies. Thank you very much. See you guys next time.